That Nicole Perna is going to do the introduction. She's here from the Department of Genetics, and she's the person that is helping to chair or chairing the UW Darwin Days event. That was Monday, was Darwin's birthday. So this is part of the Darwin Days commemoration here. And she's the person that you thank for the goodies outside. Hey, look at that.
Reception I've ever had for an introduction. After the introduction, you may change your mind. Thank you all for coming out for this special Darwin Day edition of Wednesday Night at the Lab. Before I forget, I need to thank our sponsors who generously supported bringing not one but two speakers in this week for Darwin Day and our large outreach event, which drew 190 elementary school kids on Monday. We're still recovering. But so we've been sponsored by the graduate school, by the College of Letters and Science, by the departments of anthropology, bacteriology, biochemistry, and genetics. And thanks to them, we have these two wonderful speakers and snacks. So, when Julie and John both accepted our invitation to come for Darwin Day, we were left with a bit of a conundrum. Who would be giving the Wednesday Night at the Lab talk? So, if you missed Dr. Julie Meacham's talk today in geology, all I can say is I'm sorry, and stay tuned for the next time she comes back to UW-Madison, because it was really fantastic. Julie talked about Ice Age and pre-Ice Age carnivores. It was a really interesting talk. When I went out to dinner with the two of them on Monday, I thought about volunteering to go along on one of Julie's field missions. Then today I saw the video of Julie rappelling 85 feet down into a cave, which is her field site, and I'm rethinking this. I'm still considering it, but I'm rethinking it seriously. So we had to decide between Julie, who essentially studies extinction, also known as death, and John, who studies sex. And we decided, since this is Valentine's Day, now it's debatable which of the two is more appropriate, but we decided we'd go with sex. So tonight we'll be hearing from John Logsdon, who is a professor at the University of Iowa. Although he was born in Missouri, he grew up in Iowa, and he got his bachelor's degree from Iowa State University. After that, he did a PhD in Indiana, working with Jeffrey Palmer, where he worked on what I think is an absolutely fascinating question of whether introns evolved early or introns evolved late. We won't hear about that today. What we will hear about is some work that he began, I think, as a postdoc with Ford Doolittle in Halifax, Nova Scotia, where he began to be interested in the evolution of meiosis and sex and whether the two events were coincident. So we will hear from John more about sex cells evolution, the origins of meiosis, and its sometimes strange natural history.
Thanks, Nicole, and thanks everybody for coming tonight. Actually, before I get started, I got two things. Can somebody make sure I get a snack? Because I didn't see those coming in. So I like chocolate the best. All the chocolates are gone? Oh, man. Second thing is, could I have a show of hands for anybody that was here that heard my talk on Monday? Nicole, Julie. Cool. Okay, I don't have to tell you that you can take a nap for the first 20 minutes because I'm going to use the same introduction. So, but it doesn't. It's great, actually. I think the introduction that I made for the talk on Monday is actually pretty appropriate, and there's some stuff that I overlapped with my talk on Monday. So that's great. Okay, so when we talk about natural history, of course, we're talking about evolutionary history. And So oh, awesome. So Charles Darwin, if he somehow could manage it past 200, he made it to 209 if he'd still be alive. So his 209th birthday was on Monday. Also Abe Lincoln's 209th birthday. So happy birthday, Charles Darwin. Thus Darwin Day. So what did he think about sex? Well, he grew up in a time when it wasn't really polite to talk about sex. But he thought a lot about it. In fact, he wrote a whole book about it, but encouched in this book called Descent of Man, which he talks a lot about sexual selection. So he thought about sex a lot. In fact, this key quote illustrates a conundrum he identified that he was right about, like he was right about most things, and for which we still haven't answered, which is why new beings should be produced by the union of two sexual elements instead of by a process of parthenogenesis. The whole subject is as yet hidden in darkness. The Victorian part might be in the hidden in darkness thing there too, but anyway, so Charles Darwin recognized that sex is a conundrum. More modern biologists, Graham Bell says sex is a major problem. Perhaps no other natural phenomenon has aroused. We that work in the evolution of sex often use these words, and you'll hear me here. So much interest. Certainly none has sowed so much confusion. All right, gotta love Graham. Saliato, a contemporary of mine at University of British Columbia, says, so why did so many species reproduce? So we're starting to hone in on the real question, which is why do so many species who reproduce sexually, given the cost of sex and the widespread potential for asexual reproduction? You might be thinking, yeah, that's not really an option for us, and you're right. Carl Zimmer, the famous science writer, writes for the New York Times, puts it all together. It says, today biologists understand the molecular nuts and bolts, again, pun, fairly well, but that's the how. And it's true. Actually, biologists do know a lot about the how. The why, and the why questions in biology are always the most interesting and the most problematic. The why of sex is still mysterious. Why should so many species take such a labyrinthine path to reproduction when straightforward routes are available? Okay. So let's talk about sex. Okay, what is sex? We need to define that, what I mean by it, why do it, when and how did sex evolve? I'll have something to say about that. Since it's Valentine's Day, we might ask this question. I'm actually not going to answer that question. All right, so I think about sex as sort of the yin and yang, and usually when we human beings are talking about sex, we're usually talking about this process by which gametes are getting together to create a new individual, or sometimes we're talking about how we can stop that happening, stuff like that. But it turns out that's only one half of the equation. The other half of the equation is the process by which diploid cells generate these cells called gametes that have half of our genetic contribution, right? So this process called meiosis. It turns out that this process has been studied a lot by biologists, but in fact this process is very diverse amongst even closely related organisms. So when one wants to study lots of things and compare them, this process is highly diverse. This process, as it turns out, is quite conserved, and I'll show you some evidence for that. Okay. So just to repeat that, sexual reproduction or sex is defined by the two phenomena, the gametes, which are produced by meiosis, and just a reminder of how meiosis works. You have two divisions where you double the amount of DNA, but you divide once and you have some swapping of DNA, and then you divide twice such that you get 4 cells out of this, each of which have half the genetic material. This is shown with 1 chromosome. Of course, depending on the organism, you're going to have more chromosomes. Okay, so that's meiosis, and you get gametes, you get haploid gametes, and then those gametes get together to create a cell that develops into a new organism, which is genetically different than the parents. Okay, so you have those haploid gametes, they get together. We have one from each parent, such that now we have a new individual. Okay, so it restores back to diploid, so 2 copies of each chromosome. So that was pretty complicated. Asexual reproduction is much simpler. Gametes and new offspring are produced through mitosis. Mitosis is just like Xerox copying, right? So it's just, you start with two chromosomes, you make a copy of each of them, and you divide them in half, and you get two identical offspring, identical to each other and their parent, right? So these are, one of them is sort of the mother cell. So that's asexual reproduction. There are more complex versions of asexual reproduction that I won't talk about today, but you know, just by this comparison, a lot of asexual reproduction works like this. The other thing is that life cycles for sex vary a lot amongst different kinds of organisms. So for us, we spend almost all of our time as diploid individuals, right? We're all diploids, but we have this process in our germline in meiosis that creates either sperm or eggs. Those sperm or eggs get together. They have a very short life on their own. When they get together, they form that diploid, which then develops into a new individual. Okay, so most of the life cycle is as a diploid. Some organisms, including a lot of fungi, actually spend most of their time as haploids. So they live their life cycle mostly as haploids. Occasionally they'll make gametes that will fuse, make a zygote, and immediately go into meiosis. So they spend most of their time as haploids as opposed to diploids. And some organisms, like the hornwort here, have parts of the body that are haploid and parts of the body that are diploid. Okay, and so that varies across different kinds of eukaryotes. And that asexual or growth phase can be either diploid or haploid. Okay. The other thing is that gametes, those haploid cells, actually vary also amongst eukaryotes. Different kinds of eukaryotes have different kinds of gametes. Some types are what we call isogamous. That means that they look the same, right? They have similar features. They might be genetically a little bit different. So they might be plus or minus. They might be A and alpha, or whatever, but they're essentially the same size. What we're probably more familiar with are what we call anisogamous gametes, in which one of the gametes is bigger and one of the gametes is smaller. We usually call the smaller one sperm, and we call those males, and the big gametes are eggs, and we call those females. They may or may not be motile, so they differ, so they're different. Okay. So when we think about the evolution of the sexes, we're sort of moving into the sort of male versus female category, is that we think of gametes for males as being small. There's lots of them. Their only function really is fertilization. Whereas for females, the gametes tend to be big, relatively fewer made because they're costly to make. They're not only function for fertilization, but also in most cases for nutrition. So they provide lots of energy for the first rounds of division. Okay, so that leads to this aphorism: sperm is cheap and eggs are expensive, and that actually creates interesting evolutionary outcomes in and of itself, which have been a focus of a lot of evolutionary biology, but I won't talk about. But this is just pointing out that sex is actually a really important part of biology. Sex is the rule. Most eukaryotes have sexual reproduction as part of their life cycle. It's widespread. It's predominant amongst all kinds of eukaryotes. Here's a sort of particularly nice Valentine's Day one here. Organisms basically are, most organisms have a sexual part of their reproductive life cycle. But however, lots of eukaryotes actually have part of their life cycle that's asexual. So asexuality is also very common and also very widespread. So there are fungi that reproduce asexually. There are plants. There are animals like this Daphnia, animals like this whiptail lizard. So asexuality is actually also quite common. Okay. So why have sex? In other words, what is it good for? As an evolutionary biologist, you might say, what is the adaptive significance of sex? So this is my audience participation part. So there's lots of risks and costs associated with sexual reproduction. So especially if you weren't here on Monday, I want to get some ideas about what might be a bad thing about sex. You need two to get together, right? It's a lot easier if you just got one, right? That's a cost. Yeah.
The energy used in attracting a mate.
Exactly. So it might be costly. So not only you have to find one, but you got to spend some energy to find one. Yeah. Anything else?
competition for mates?
Competition for mates, yeah. Another form of energy use, yeah. Anything else come to mind? Yep.
You don't know if this new combination is actually going to be successful.
Right. So you're shuffling the deck, right? So, you know, if I'm obviously the perfect human being, I should just make a copy of myself, right? You know, why would I mix up my cards with another individual, right? So that's that. Yeah. So that potentially is a cost. In my case, maybe it's not a cost. I don't know. All right. So those include searching for a mate. Got that one. Requires time and energy. Peacocks, you know, hey, you know. All right. That mate might require some investment. Food, territory, defense. Sure, you love each other, but is your credit compatible? That's a very millennial version of this argument, right? We didn't get this one, but obvious, you know. So getting together of two individuals, I mean, we transmit the flu from person to person. We transmit worse things during sex, and actually, all organisms have sexual transmitted diseases. Plants actually have them. So it's just a consequence of that proximity that has to happen. So yeah. And finally, there are some actually really dangerous things, like in some organisms, like these praying mantises. And for those of you who are laughing, already know what's going to happen here. Is that he's going to mate with her, and she's going to turn around and literally bite his head off and eat him. Okay, cannibalistic sex. Okay, so that's an extreme version, but you know, sex is complicated and it's maybe dangerous. Okay. So why do it at all? I mean, many of these organisms can reproduce asexually. So what that means is, John Maynard Smith, my last evolutionary biologist, making a quote here, says the evolution of sex is the hardest problem in evolutionary biology. And what he really is thinking about is that in populations that can reproduce both asexually, they have a choice, if you will, asexually and sexually. Why doesn't asexuality take over? And part of the answer to this question is emphasized by this idea that in a sexual population, if you're a female and you on average have to produce Oh, thanks, Java. That's great. I'm going to skip that version. Thank you very much. Wow. Okay. In a sexual population, if you're a female, on average, you have to produce a male and a female. Whereas if you're in an asexual population, you're going to produce two offspring. You get to produce both that are female, right? That each get to turn around and turn. So males are kind of useless in this population growth business, right? Because each female has to produce half of her offspring being males, and the only utility of the male is to generate offspring. He can't carry the offspring himself, right? Tough for us males. Okay. All right, so this is how it works out if you think about it. So these are snails. I'm going to get at the very end. I'm going to talk about our research on snails. This slide comes from my colleague Maureen Nieman. But in an asexual female, she gets to produce two females. A sexual female produces a female and a male, and you can see that fairly quickly the population of asexuals goes way up because each one of them can produce two females, right? So that you can see that it outpaces the population growth of a sexual. All things being equal. And you can see that even when the asexuals start out very few, they can basically, after a few generations, completely overtake a sexual population. So asex really works. It's really super powerful. So what that must mean, then, why aren't most organisms asexual? Asexual reproduction should take over, but it's not the case. Most organisms, as I said, reproduce sexually, and both sexual and asexual modes are used in many organisms. So that means that sex must confer some benefits that overcome advantages of asexual reproduction. So what can those benefits be? And of course, evolutionary biologists have spent many years thinking about what these benefits could be, and we have basically a lot of theories about it, some of which are supported by different kinds of data. But for me, the three theories fall into three categories, and I call them the good, the bad, and the ugly. Okay, so the good might be the sort of thing that you might think of is sex must be good. Sex increases variation, it's beneficial for adaptation, and this guy, Weissman, actually was one of the first to bring forward this idea. Or maybe, in two different individuals that have good mutations, when they have an offspring, they can bring those good mutations together. That's attributed to Ronald Fisher, and I'll show how that works here. So this is a sexual, right? So you're in a population, and the gene A gets born, it's a good gene, and then you have the gene C that arises, and it's a good gene, and B, but as long as these are all getting together and recombining and mixing it up, is it fairly quickly you can get a really good genotype which has A and B and C all at the same time. Okay? If you're an asexual, it doesn't really work that way. You have to wait around till the A mutation arises, and B might arise and go extinct. But in the A background, then you have to wait for the B mutation to come around. And in the AB background, you have to wait around for the C mutation. So the idea is that these beneficial mutations aren't able to spread in the population as quickly. Okay, so that's the Fisher idea. So then the flip side of that is the bad, which is that if you're an asexual, actually you have a really hard time getting rid of mutations that you already had. And that's attributed to this guy, Hermann Muller. And here's a visual of kind of how it works. In an asexual, we have a bad mutation. It's passed on to all offspring forever. So let's say we have this C star, and we have a mutation that's missing this arm in this lineage. And from that point forward, all the descendants of this lineage also have a missing arm. And in this lineage, another mutation happened, a PowerPoint-mediated one that kind of makes it a pointy arm. And all the subsequent ones are going to have a missing and a pointy arm, and you can see. So actually, the Muller's ratchet is actually, I know this audience will understand photocopying. My undergraduates don't understand photocopying anymore, but if you take a photocopy of a photocopy of a photocopy of a photocopy, that's Muller's ratchet. Okay, so the bad stuff just accumulates over time. Okay, I can't use that in some audiences. Trust me, they don't know what photocopy is. All right, so the last one is the ugly. And the ugly is particularly interesting.
Your audio just went off.
Oh, well. My audio went off, okay. How about I turn it off? Back on. Funny. Batteries, maybe? I can use the one.
Hello?
All right, you've done enough loudly. Good. Thanks. No problem. This is very minor in the various kind of things that can happen when you're giving lectures. Okay. All right, so the last is the ugly. And this is the idea that variation generated by sex might particularly help you in changing environments. This is attributed to Bill Hamilton. Okay, and the idea here is that you might have this purple snail has again from Marie Nieman, who loves the snail slides. Genotype with poor resistance to dry conditions. Well, it's in a wet condition. It's doing really well. In fact, it's doing super well that these other genotypes aren't around in this wet condition. But when things dry up, then this genotype dies. But this genotype this population is diverse and so can persist. So you can see that sex in this kind of situation might be good. Well, environments in general, we think of these abiotic environments, we generally think of them as kind of relatively slow changing, and biology can keep up with them. I mean, that's what the administration thinks about global warming. But in any case, abiotic environments are potentially slow changing, but actually, environments in which biology exists are actually with other biology, which is evolving with it. So what if the environment itself is evolving, right? So you're evolving with parasites or pathogens or predators, right? Which are themselves biological and moving fast. And so the idea is that sex might be good in situations where the populations are kind of evolving with each other. And it's come down to this description that has been analogized to the Red Queen in Alice in Wonderland, where the Red Queen says to Alice, now here you see it takes all the running you can do to stay in the same place. So in other words, as the host changes, or as the parasite changes, the host has to change. As the host changes, the parasite changes. So it's a tit for tat, in a cycle, and sex would help that, right? So if you're in one genotype, the parasite's going to figure you out and decimate you. So those are the hypotheses and evolutionary biologists think about with regard to what we call the maintenance of sex problem. So this is the sort of end of the beginning, which is that that's just to sort of frame this up, which is sex is super common. Okay, it's extremely common. It's costly. We talked about some of those costs, and that I didn't mention, but these asexual lineages, as it turns out, we know are actually failures. So long-term asexual is not a good strategy. So that must mean that sex is associated with profound benefits. Is that sex must be good for some of the things we talked about, or others. Okay. All right. So that's sort of the general framework about the evolution of sex. And now I'm going to tell you about my own research in my lab about the evolution of meiosis, which is half of that equation. So looking at the evolution of meiosis, so the big questions that drive my research, and I'll tell you a little bit about some of the research itself. So the big questions are, when did it arise? Okay. When did this process called meiosis arise? How did it arise? And where did it come from? From what prior functions did it arise? Given that it exists, I'm interested in how it's evolved, right? So, how has it changed? How, what happens to different organisms over time? One important question is that I've focused on a lot of attention and energy on is it possible for meiosis to be lost? In other words, remember I told you that asexual lineages tend to not persist. Does that mean that meiosis as a process has some role in that? Is it possible to lose this alternation of haploid diploid life cycle? And then all the time I just spent talking about these sort of maintenance of sex ideas, what can the evolution of meiosis tell us about those kind of questions? Okay. So first thing to do is to ask who is doing it. So among organisms on the planet, what do we know about which ones are actually capable of doing meiosis and which ones are not? So one thing we do know is that prokaryotes don't do meiosis. No prokaryotes do meiosis. They divide by another process. They might have sex by a kind of one or a few genes at a time kind of business, but none of them do the wholesale shuffle the deck kind of business that meiosis is. So when I started looking at this question, I wanted to say, well, over the tree of eukaryotic life, what's known about the presence of sexual reproduction and meiosis, or meiosis? And so this is a phylogenetic tree, if you will. It looks a lot like a comb. Some things are probably familiar, so animals and fungi, and there's some plants here. But most of these things on this tree are actually unicellular eukaryotes. And two things about them is that we don't know their exact relations to one another, for the most part. The other thing is that we don't study them a lot, but they do represent a lot of diversity. Some organisms we think we know don't have sex, some things we do, but there's a lot of unknowns, right? So you don't get a lot of talks on protist biology in this series or any other public series because not that many biologists work on these organisms. So there's a lot of unknowns. So the evidence for sex comes from sort of two kind of categories. One is people that have gone out and done population genetics, where they go out and get individuals and they have some way of assessing whether they're mixing up their genes or not. So that's population genetics, or catching them in the act, direct observation. You can either go out and see them in the act in the wild, or bring them in the lab. But the problem with these two ways of looking at it is that it creates a lot of like, oh, I didn't see anything. Okay, and so it's absence of evidence. So when you have absence of evidence, how are you going to infer what's going on? Is it asexuality, or is it just that they're a little bit shy? Or they don't do it very often? Or whatever. So we have to think about whether we know whether it's a question mark or really a minus. Right? So the absence of evidence is not always evidence of absence. When I started this work, we had a view of life that kind of looked like this. And this is a phylogenetic tree of life. It's constructed using a particular molecule. And it groups things in a nice way. There are two kinds of prokaryotes, eubacteria and archaebacteria. And eukaryotes are all shown here. and the familiar animals, plants, and fungi. And then these are all those these branches represent all those single-celled eukaryotes that most people aren't super familiar with. When I started thinking about this, there were a few of these things that seemed like they were the first branches here. So there's animals and fungi, late branches, and then these things seem to diverge early. Microsporidia, trichomonads, and diplomonads. Giardia. Meet Giardia. Hope you haven't met him before. Somebody laughed. Okay. Okay. But in any case, what was interesting about these things is that they weren't known to have sex. Okay. When I first started working on this back in Halifax, they weren't known to have sex. So they represented maybe a group of eukaryotes that were bona fide eukaryotes, but they hadn't acquired sex in meiosis. So that was pretty intriguing. And we called that the meiosis late hypothesis. So meiosis was a feature that derived within eukaryotic evolution. Well, we've gone from this highly resolved picture to actually this is a kind of, even though it's 2004, it's a modern view where it's not really clear what the earliest branch is. In fact, we have animals and fungi, and we have plants that all kind of go down to this early common ancestor, and then there's kind of this explosion. So all those groups of individual single-celled organisms are sprinkled along this tree. It led my colleagues to generate this hypothesis that this last common ancestor, so here are the prokaryotes, and here's the last common ancestor of eukaryotes here, would have been a complete eukaryotic cell. It would have had a nucleus, definitive of eukaryotes, and some other things, ER, Golgi, and then underwent mitosis and meiosis. So that was just an assertion on their part, and I'm going to show you the data that indicates that is true. Back to this ribosomal RNA tree, just to give you an idea. Those are those early branches here. This is that same thing. Those are Giardia and Trichomonas down here. But this is just to show you where we sit. So we're up there with our fungal friends. Okay, so animals and fungi. And the reason why that's important is because what do we know about this process of meiosis that I'm interested in studying? Almost all of it comes from that purple box, which I've exploded here to be fungi here and animals here. So we almost everything we knew about meiosis, certainly 10 years ago, and even continuing, comes from things like brewer's yeast and maybe a few other fungal groups, but from our favorite Drosophila and C. elegans and us and mice, which still represents a small slice of eukaryotic diversity. We know a lot about how meiosis works from those organisms. Increasingly, we know something about how meiosis works in plants, which is great, actually. But what I really wanted to do was to know about how meiosis works across all of eukaryotes. I want to be able to take what we know from here and spread it all over the tree, right? So the nice thing was, at the time I was setting up my own lab, was the time when people were really earnestly starting to sequence entire genomes from things other than us, right? So from a variety of different organisms. And that's shown here. So this is that same tree that I showed you a minute ago, but it's just sort of splayed out here. So here's animals and fungi, here's some protists, here's some plants, here's some more protists, here's some more protists, etc. Okay? So there's animals and fungi and plants in the sort of bluish boxes. Those are the organisms that we know how meiosis works and some of the genes that are involved in the process. The boxes represent about the genomes that were available at about this time, 2005. So we had a genome or two pretty well spread across the tree, but over the years, those genomes have been increasing over the tree. So there's 2011, and that's last year. So now we actually have entire complete genomes, the set of all of the genes in all of the major branches of eukaryotic evolution. And so what we can do is we can take and find genes that are present in animals and fungi and plants and look for their homologs, that is, the similar genes in all those other organisms, okay? So, and it turns, and I'm going to tell you a little bit about how we did that. But first I want to remind you a little bit about how meiosis works. I already introduced it, but this is my sort of University of Iowa version. Meiosis is has this set of two divisions. The first thing that happens is that, just like in mitosis, you replicate the chromosomes. So this cell has four chromosomes. Actually, it has two pairs of chromosomes. Okay, So this cell got the black ones from, say, mom and the yellow ones from dad, let's say. Sorry, I have this, I told you wrong. The yellow, yes, that was what I wanted to say. Even it's confusing to me sometimes. But what happens is they replicate, but the neat thing that happens in meiosis is they don't just separate and divide up like what happens in mitosis. They actually pair up and they swap parts. Okay, and then they go through another division where they divide in half. Okay, so notice these gametes actually have 2 chromosomes instead of 4. Okay, turns out all the neat stuff happens in meiosis 1. All these steps here where they're pairing up and they're swapping parts. The biological mechanisms, the molecular mechanisms that happen at this step are essentially mitosis. Okay, so the machines that do this we already have because we already have mitosis. A little bit more about that. So here's mitosis. You just replicate the chromosomes and then split them in half. Whereas here you replicate the chromosomes, you pair up, you swap, you split them in half, and then you split them in half again. Okay? But what's neat here is what's happening here is this recombination. This is that part where different parts of the chromosomes are swapping with each other. And there's a whole bunch of genes that encode proteins that do this stuff. Okay? So it's the evolution of the genes that do that stuff that I'm interested in because they tell us something about how that came to be. Here are some of those genes, and here's a picture. If you're a are there any biochemists in the audience? All right. So anyway, but this is a picture of what's going on in somewhat more detail. You don't have to really pay that much attention, except for to say it's complicated. There's lots of steps, and there's lots of players. These are the genes that are named here. The green ones, which you don't have to look at their names, the green genes are genes that are only involved in meiosis. They're specific to this process, and their only functions are to do this thing in meiosis. There's a bunch of other ones that have other functions in mitosis, let's say. But there's a whole bunch of stuff going on here. So there's a variety of genes to study their evolutionary history. So how are we going to do that? I've already given you a hint. The criteria are to find ones that we call are meiosis specific. Their job is only in meiosis. They have to be conserved enough to study. I sort of hinted at this, but I'm not going to tell you what this means. Eukaryotes as a group are about 2 billion years old. Okay? Our divergent from a common ancestor of prokaryotes is about 3.5 billion years old. So the genes that we want to study, if I want to study the whole of eukaryotic evolution, I have to be able to study genes that I can identify that have been conserved enough that their sequences I can identify over 2 billion years of evolution. Okay? So they have to be conserved enough. And in fact, many of them are so conserved that I can identify their similar homologs in prokaryotes, 3.5 billion years divergence. Okay? I'm going to tell you about the duplications in a minute, but it turns out that many of these have evolved, we can figure out have evolved from genes that do something slightly different but similar. So I go into this project and I say, okay, I am going to decide how this is going to work, and I'm going to find these genes, and all the species that are known to undergo meiosis will have the genes, and all the species that won't have the genes. That's how it's going to be, right? Unfortunately, biology doesn't always cooperate with these things. Is that evolutionary biology is interesting. So this is a sort of best case scenario that turns out not to always work out, but it's a good starting point. Oh, that's great. Sheesh. Oh, lovely. Wow, I apologize. This is something today. All right. What I will say is that right before this, I was messing around a little bit with my talk, and the thing crashed on me. So sometimes Microsoft just is not cooperating. Here we go. Okay. So this is an example of those ones that are doing related jobs. So this is that same kind of diagram that shows how DNA gets processed and recombined over here. These are names of genes that are present in a particular species, yeast, that we study a lot, lots of people. There's a bunch of genes that are used both in meiosis and also mitotic processes, and there are ones that are these meiosis-specific ones. And these are just the gene names. And what I hope you can see is that is there's a bunch of them here, but there's a bunch of them here too. So we can study the evolutionary histories of all these genes. And it turns out that some of these are generalists. They're involved in general processes of DNA repair, and some of them are specific. And the ones that are the same colors, they have evolved from each other by duplication. So they're similar to each other in that they share a common ancestor gene. So that's happened a bunch of different times during the evolution of meiosis. So it already says that the genes that are involved, that are specific to meiosis, already have arisen by duplicating another function that was close to it and pulling it into this very specific process. Okay, so that tells us already. I'm going to show you some more data in a minute. But all right, this is your eye test now for the next 3 or 4 slides. This is actually what we do. I mean, I'm actually eliminating lots of detail, but what we do is we go through genomes. So these are species and indicating genomes, and there's genes along here, and we go through and we ask, are the genes there or not? I'm not going to bore you with the details of verifying, but we do careful analysis to make sure. So some genes, like this one called SPO11, everybody has. Okay, that's a meiosis-specific gene. Other ones, they're meiosis-specific, and it's a little washed out, but you might be able to detect the yellow columns a little bit. But some of these actually, genes like HOP2, MND1, DMC1 are missing in organisms like Drosophila. We know that Drosophila has sex and meiosis. So it already violates the criterion. It's capable of meiosis, but it's missing some of these genes. So that happens. So we have to be a little careful of that. So that was one thing that we were aware of, is that, yeah, it would be great if all of them look like SPO11, but stuff happens in evolution, right? So in flies, they've lost some of these things. So you can't just use one gene as a marker. First thing we did was we went after that putative early branch thing called Giardia, and we asked, does it have the meiosis genes? And it does. It's got this one and that one and that one and this one. Oh, it's missing these two. Oh, that was interesting. So remembering that, okay, yeah, it has most of the meiosis-specific genes, but it doesn't have this one. We made the strong inference that actually it probably is capable of doing meiosis and sex. And remember, it was one of those putatively early branches on the tree. So we published this result, and then later on, some people went and did those population genetics work to show that, in fact, those populations were exchanging DNA. So that was kind of cool. Okay. So then we keep moving on the eye charts. We're adding more genes. We're adding more species. In this case, we added this thing, which is another one of those early diverging lineages, Trichomonas. It has all the ones that Giardia has, plus it has a bunch of other ones. But you can already begin to see that this thing is not all filled in. So instead of pluses now, it's just color, filled color. So already it's true that not all organisms have all the genes, but most of them have most of the genes. Okay, and in certain organisms they got all the goods, right? So we do, and yeast and Aspergillus have all of the genes, which means that those genes were in the common ancestor, and the missing ones are that have been lost. Okay, so that's the inference. Turns out that this thing Trichomonas is probably doing it too. And remember, I said Giardia was missing these. Trichomonas has them. So those genes are probably present in the common ancestor of these guys as well. More eye charts, but this is just for fun, just to say, okay, more genes, more taxa, and that's what we do. Okay, so we're just generating more and more and trying to look for these patterns of presence and absence and correlate them with the evolution of certain meiotic novelties. Okay. This is a slide I didn't show on Monday, but I thought I'd put in, which is sometimes certain processes, and you might not be able to see this as well as I'd hope, but sometimes certain processes, so some genes might be able to be lost. So like here, these are gene names that you might have already remember. MSH4 and 5 might be lost, but there are overlapping pathways. So in some organisms, both pathways exist, and in some organisms you can lose one or the other. So that's one thing that we've seen, is that there might be some redundancy in some of these processes. The other thing that we started doing was, again on the eye chart phenomenon, is to try to create a more general picture of the evolution of meiosis across all eukaryotes, and now these are the same colors of those big groups, and now we have, in this analysis, we have at least one species for every one of those major groups. And we can then combine them together. So if it's present in at least one species in that group, then we call it a plus. And then we're trying to figure out whether we don't really know what the phylogeny, what the order of branches are in the tree. But if we've covered the entire tree, it doesn't matter where we know where the deepest branch is because everybody's got it, right? So that's what's shown here. If we do that, we just count up, at least 10 of these genes are present in the common ancestor of all of them, just by simply saying, does at least one lineage have them? But then we have this other trick where it turns out many of those genes have evolved from each other as duplicates, as I've indicated before, such that maybe it's lost in a particular species, but its presence in this species demands that it be present here because its common ancestor is back to here. And this is how gene duplication helps us to make this really strong inference about the evolution of meiosis. So when we do that, we can trace all of these genes back to the common ancestor of all eukaryotes. So it's a kind of evolutionary trick that we can use in the analysis, but it's not a trick with regard to how it works, is they had to have had those set of genes in order to give rise to the current distribution in eukaryotes. So we don't really need to know who the deepest branch is to say meiosis is in fact at the present at the common ancestor of all eukaryotes. So that's just a summary of all the data. I'm not going to belabor that. It recovers a tree of eukaryotes that's consistent with what we know. And we learn that many of those genes have evolved from gene duplication one to another. Here's an example. These two genes, this is a generalist and this is a meiosis-specific one. Here's a generalist and here's a meiosis-specific one. Here are two meiosis-specific ones, but here are some generalists and here are some meiosis-specific ones. So those new functions that are meiosis-specific didn't come out of nowhere. They came out from genes that already existed. Many of them actually in prokaryotes. Okay? So that's shown here. I'm not going to belabor that. So that was kind of the big picture. So we were able to see that big picture of those meiosis genes across all of the tree of life. So what I wanted to do next was to then say, well, what's going on at a more fine scale, and a fine scale being a whole kingdom of eukaryotes, fungi, and so we chose fungi to look at more carefully, and so we did the same kind of thing. I'm not going to belabor this kind of data very much, except for to say we had some more species and more or less the same genes, and we can start to see patterns that, for example, if this gene is missing, that gene is also missing. If this gene is missing, the other ones are missing. Turns out these two work together in a 2-protein complex to do its job. These work together in 3 proteins at a time. So if you lose one, you might as well lose them all. Okay? So that's something that's going on. So this is, again, more eye chart business, but to point out that more of what I just said, but we can also catch genes in the act of getting lost. So we can find genes that are that their dead bodies are clearly visible in the genome, but they don't encode a functional protein. We call those pseudogenes, so we can catch genes in the process of being lost. So we ended up looking at lots and lots of species. Our winner so far of organisms that have the fewest meiotic genes is this thing called Malassezia. And Malassezia, are there any mycologists in the audience? Okay. Does anybody know what Malassezia is? Malassezia is the fungus that causes dandruff. So the dandruff fungus has the record for having the least number of these meiosis genes, but it still has some of them. So is this a case of an organism that's capable of losing meiosis? I don't know. It's not studied by lots of people. Okay. Even though probably lots of people wish it weren't on them, I guess. I'm going to skip this. Okay, so just to sum up is that machinery, those genes that are involved in sex, evolved once, early. All eukaryotes have them. There's lots of duplications. There's lots of gene losses. And that anything that we thought was asexual, and remember we thought that maybe Giardia and those things might have been asexual, we now think that they are derived from sexual ancestors. So sex and meiosis arose early. Anything that maybe is asexual is a result of loss. Okay, but what about everybody else? Okay, there's lots of other organisms out there to think about, and can we use what we know about that natural history as a way of testing an organism that we want to know about its sex life? So can we use this inventory kind of way of thinking as a way of going and looking as a toolkit? So the idea being that a presence or absence of one gene is not enough for a strong inference. We have to have the set of genes I told you about. There's lots of asexuals out there in biology that people have claimed are asexual, long-term asexuals, etc. And we really want to ask this question, is everybody doing it, right? Is it possible to lose sex? Is sex required of being a eukaryote? So here's the approach. We call this CSI Meiosis. We're going to use some exemplar biological species. SpongeBob works pretty well because when I looked it up on the web, his creator says he is asexual, which already makes you wonder. Maybe if it was Sponge Jane, I'd be a little bit more believing of it. But anyway, there's SpongeBob. But now we're going to ask, is SpongeBob sexual or not? Come on. Okay, so prior to the meiosis way of looking at it, we might go and look at SpongeBob populations and ask whether there's recombination or there's swapping of genes. But you might imagine that's not what I'm going to do first. I'm going to go grind up SpongeBob, sequence his genome, ask if the meiosis genes are there. If they're there, I might go back and ask for recombination. How many? Which ones? Is it just like Malassezia, only one or two? Or is it like most organisms that have a whole set of them? Because if it's got the whole set of them, we're pretty sure that SpongeBob is doing something, okay? Are they expressed? Are they turned on? When and where? We know something about how meiosis works. We know that gene A is turned on before gene B is turned on before gene C, and we know that they're expressed at in one place in the organism. Maybe the germline for SpongeBob is up here in his ear, and those genes should all be expressed here, and maybe not over down here in his toe. Something. We can potentially assess their functions in a kind of genetic way if we want to. Because in the end, what we really want to do is say, this thing was thought to be asexual, but we can figure out how to get SpongeBob to go through meiosis, because then he's not an asexual, and most kids won't want to know what that looks like. So maybe even adults. All right, so that's the idea. So we would solve that case. So our SpongeBobs are these organisms. They're actually the Sponge Janes. These are called Deltoid rotifers. So these are microscopic animals that live in water, and these are mostly freshwater ones. And these are all females. Okay, these are all females. There's a mom with her soon-to-be daughter who's going to be identical to her, and these are their closest relatives. And these are called monogonate rotifers. And these alternate between asexual reproduction and sexual reproduction. So these are thought not to have any sex. Just a little bit more about them. They actually are thought to be obligate asexuals. They're part of another group where they're clearly derived from sexuals. They don't have males. There's no known meiosis. They form a single lineage, a single evolutionary line. They're old. They're 100 million years or so. They found them in amber, so they're pretty good dates. They don't have homologous chromosome pairs. Remember, that's sort of a hallmark of eukaryotes, is that you got a mom chromosome and a dad chromosome, and they're similar to each other. They don't have a certain kind of transposable elements, and they have what's called allelic heterozygosity, or the Mendelson effect, which I'll explain here in a second. So these are all things that were consistent with them being asexual. Here's the Mendelson effect. So here's a tree of four different species, and they split here, and then there's a split here. What happens in a sexual species is since you got two copies of every gene at every locus, you have an A, in this case, an A version and a little a version. But over species time, they're actually recombining with each other. And if we go into our genomes and we look at our allele that we got from our mom and our allele I got from our dad, they're almost identical. They might be 1% different. So they're mostly the same. But if we then compare our alleles to chimpanzees, our closest relatives, they're going to be many more percent different than that. I should say 0.1%. Sorry. Yeah, about 1 in 1,000-ish for humans. So, but what happens in the absence of sex is that these things are no longer recombining. They're not exchanging, so they get frozen and fixed in time. So once you lose sex, then your big A allele and your little a allele are becoming more and more and more and more different, and the more different they are, the longer time they've been asexual. That's called the Mendelson effect. And so that was purportedly a signal that people could look for and found in deloid rotifers. The problem is that you can get the same thing that looks similar to in genomes that have doubled the genome size by virtue of what we call polyploidy. So if you have an extra set of chromosomes that duplicates early, they can end up looking like this so-called Mendelson effect. And it turns out we figured out that deloid rotifers have done this, unfortunately. So that kind of evidence went away. So we went looking for the consequences, which was looking for these meiosis-specific genes. And the idea here was that if they were present, they would very likely be sexual. If they were missing, they would be consistent with ancient asexuality, which was the preferred hypothesis. So we did a lot of work, and this is summarizing all of our work except for this line here. We did a lot of work looking for these genes, and it turns out we found some of these genes. So here's our one that I mentioned before, MSH5. It's present in these are the deloid rotifers. The green boxes say it's present. We found this thing, MSH5, also meiosis specific. The pink ones are meiosis specific, but we didn't find some other ones. Then this genome came out and sequenced the whole genome, and they found the ones that we found, plus they found this one and that one, and they didn't find these 4. So now it's a tie. 4 missing, 4 present. But yet they have this title, Ameiotic evolution. So this is kind of interesting because I think they haven't proven that it's ameiotic. In fact, their argument is pretty strong. They say that there's positive evidence for asexual evolution. When you look at its genome structure, it doesn't allow pairing of homologous chromosomes, and therefore seems incompatible with conventional meiosis. In other words, they can't imagine that meiosis could be happening, given that when you look at the chromosomes, they're all scrambled. So these are just letters to show how the chromosomes would be scrambled. And so it's the argument from incredulity. Okay? Not my favorite kind of argument, but it's put forward. And so that's where it stands. These meiosis-specific genes are there. Maybe that says they have a capacity for meiosis. Maybe those genes have different functions. There's 4 of them. They'd have to acquire different functions for all 4. And one possibility is that they got them from outside. I think we have eliminated that possibility, but it's still a formal one. So this continues. So now, more recently, people have been looking in these deloid rotifers and said, well, yeah, what does this mean? We're finding some swapping of genes among deloid rotifers, and this paper comes out and says, well, actually, they're horizontal transfer, and it looks like there's meiosis happening with other deloid rotifers by movement of DNA that they're picking up from their environment, and they actually coined a phrase called saphomixis. In other words, they're having sex with their dead relatives. Okay, their dead relatives have genes that are of slightly different variety, and they're just picking up one gene at a time. Kind of a little bit like bacteria do it. So that's the idea. And then the thing is claimed to be ending. But as with all kind of good science, there's controversy and there's disagreement, and so we get to go back to the lab and do more work. Okay, so with all that, I'm gonna wrap up and say what are the answers to some of the questions that I posed. When did meiosis arise? Very early in eukaryotic evolution, prior to eukaryotes as we know them. How did it arise? Well, it's pretty clear that most of the genes have a story in gene duplication. So there were lots of gene duplications, maybe one big round of gene duplication by whole genomes. Where did it come from? I didn't talk much about this except for this. I pointed out some of the names of some of the genes. So a lot of those genes are involved in DNA repair. The ancestral state is handling DNA. Well, that's not a big surprise since meiosis is a DNA handling problem. How has it evolved? Well, there's been a lot of gene loss. You saw a lot of that. There's been some gene gain. I didn't tell you about that, but certainly more duplication. So although the process of meiosis is highly conserved, there's lots of modification happening over those 2 billion years of evolution. Can it be lost? I'm skeptical about the dolly rotifers. Others are less skeptical. I think, like I say, good science leaves open questions. And I'm not going to be able to really tell you about the consequences because I don't know what they are. But I think that those big questions that I set up early in the talk really are the ones that are more general. And I'm hoping that this meiosis evolution will tell us something about sex itself. Okay, so I'll end there and thank you and invite any questions. Thanks. One moment while I put my acknowledgement slide up. Yeah, so the work that I talked about is done by these people, and yeah, shown here, and thank you. Okay, questions? Yeah.
So going back to what you were saying about environmental change affecting this kind of thing. I mean, I've had some limited experience with C. elegans.
Okay.
And I know that there are like environmental cues that can trigger more males to be in the population than normal. Is there a way to model kind of what that percentage has to be in populations that can reproduce either way?
Yeah. So great question. I can't answer precisely for C. elegans, but it gives me a chance to answer in a way that it turns out that in the case of environmentally determined sexual reproduction, that's actually for organisms that aren't required to go through sexual reproduction in order to reproduce, like us. So it's fixed in our life cycle. The ones that can go either way usually have an environmental cue to go sexual. And this is a great thing for Valentine's Day. Almost all organisms have sex when things are bad. It's not like, hey, honey, I got a promotion today, or whatever. It's like, oh my God, I lost my job, or I've run out of food. And so, in the case he's talking about is C. elegans, where these are hermaphrodites mostly, and they are mostly having sex with themselves, truly. They can actually reproduce and create males every once in a while. And so the conditions in that case to give rise to males is some environmental trigger, which I don't know what it is. In the case of these monogonate rotifers that alternate between sexual and asexual, the cue to go sexual is crowding. There's too many of you in here. We're running out of food. We're going to go sexual. So almost always there's some kind of environmental cue to say it's time. That things are the good life is done. We have to escape and do sex. So sex is kind of like the escape valve in most of these organisms. I'm not sure that answered your question, but people have actually modeled that mathematically, etc. And like I say, it's one of my good favorite questions because I got to say that what I said. Yeah, over here.
Me?
Yeah.
Oh, okay. This is pretty basic, probably, but when we're talking about the asexual species. Can we say they clone themselves?
Yeah, absolutely. I could have used that word interchangeably. Yeah. And in fact, our species could technically go asexual, but it requires technology to do it. We are not biologically set up to reproduce asexually. Although the case is that many organisms that reproduce sexually by rule occasionally spit off asexuals. So you're probably familiar with like the asexual sharks and things like that. So occasionally in zoos, you see these asexual things. They tend to not be able to persist very long, but there might be these propensities that are around that one can reproduce asexually. It's always the females that can do it, of course, because we're kind of stuck with our limited resources of our gametes of males. Yeah.
So for mammals, I think, and maybe others with sex chromosomes, X and Y chromosomes, are the meiotic genes located on those chromosomes or more broadly throughout the
Yeah, that's a good question. So I didn't talk about sex chromosomes at all, but yeah, so sex chromosomes are part of a more general theme of what we call sex-determining mechanisms. And remember I showed early on, even the things that are isogamous, you can have pluses and minuses, and those are kind of males and females. And they aren't always determined by a single chromosome. Sometimes they're determined by a single gene. And what those do are, and in our case, there's one important gene on male Y chromosomes that say male and determine a whole bunch of downstream genes. So all the meiosis genes are spread around through our genome, and females and males might deploy them slightly differently. In mammals, there are sometimes, meiosis works different in boys and girls, but the genes that do the nuts and bolts stuff are generally the same ones. They might be regulated by whether it's a male regulation scheme induced by the Y and SRY and all that stuff, or female. Okay, so sex chromosomes, determine the fate of the germline being male or female, but they don't directly control meiosis per se, because that happens irregardless. Yeah. Good question though. Yeah.
So it seems like asex would be beneficial to have. Why did it go away?
That is a great question, right? So lots of organisms have dispensed with the ability to alternate between generations. I have thought about that question, but it's late at night and I don't remember what I've thought about it. Anybody got some ideas? So I think some of it is constraint, right? Is that there were trade-offs between being able to do that and being really successful with one strategy. And I think there's a lot of stuff in biology like that. It's just that for whatever reason, one ended up getting fixed. I think multicellular eukaryotes often will set aside their germline, right? It could be that it's difficult once you sort of developed further and further a body plan that includes your germline in some way, that it might be really hard to reproduce in a way that you've tried to that evolution has selected to go in a particular way for outcrossing, let's say. I think I'm mostly just thinking out loud, but I think that's a pretty good question. It probably has happened multiple times as well. So it's not just mammals, for example, that are stuck, but there's other lineages that basically have given up alternation between sex and asex. But yeah, let me think about that some more. Thanks for a thought-provoking question. Yeah.
The genome is an information storage system. How close are we to our limit of being able to read it and pack it back?
So the question is, if the genome is our information storage hard drive, if you will, can I make an analogy? How close are we to our limit of gigabytes in our hard drive? Is that a reasonable way of rephrasing the question? How close are we to our limit of being able to have that information? I don't imagine that any organism is. I mean, I think there's this trade-off between having enough DNA to encode the information and do all the things that you do, but the fact is that for us, we only about something on the order, and it's debatable amongst people, about 5% of our genome encodes the nuts and bolts stuff. It's actually chock full of other stuff, mostly selfish DNA that replicates on its own, among other things. There are people that argue about the percentages, but we're able to and other genomes are much bigger and build perfectly fine organisms, and they're even more full of junk. So I think the thing about that raises the ceiling on our ability to have bigger and bigger genomes is the mechanics of transmitting it from generation to generation. It's not our genome can hold, by that argument, could hold 20 times more information, and our genome is certainly not even close to the biggest ones on the planet. So in the end, I don't think that's the problem. It's how to do the mechanics of transmitting it from generation to generation. And selection would act to reduce that if it becomes a problem. Yeah?
Well, to follow that paradigm, could you comment on a paradigm of poor coding and massive amounts of lines of code versus a small virus that can totally bring down the millions of lines of code, and how evolution follows through with that to get better large coding, such as the human genome?
So, but I think the analogy is that it's actually pretty crappy coding. It's interspersed on the whole hard drive in different sectors, all spread apart. But its evolutionary history has retained the connections from this sector to that sector by whatever means. That it's not. If you like the coding analogy, it's not annotated very well, right? And one thing that can mess up the code, it would be analogous to a virus, would be an actual virus or a thing like a transposable element, which can, and we know that can happen in some organism where you get a piece of infectious DNA and it moves around the genome and causes havoc. But cells have ways of dealing with those things, kind of like antivirus software, I guess. But I don't think we're limited by that. I think the code has been, there was code written at the common ancestor of life, and some of that code has been maintained. It's been being added to and there's been this continuity of life over the whole 3.5 billion years of life on Earth. I think I'm trying to read that code in some ways, and other people others. But I think it's not totally straightforward. How it's been packaged in the genome all the time. Yeah. Yeah.
So this is both a why question and then maybe a future of meiosis question. But it's my understanding is that in mammals the Y chromosome has only maybe less than 5% the number of chromosomes as it once did.
Genes. Genes. Yeah.
Genes as it once did. And as those chromosomes do include the X chromosome. So in order to have meiosis, you have to have fertilization, and it seems like the Y chromosome necessary for fertilization is getting smaller and smaller and smaller. Is it going to keep getting smaller or to go away? Could that be the end of meiosis?
So the thing is that you're absolutely right. So the original Y chromosome was an X chromosome. Okay, so the sex chromosomes in us used to be two X chromosomes. They weren't called X chromosomes; they were autosomes. But that sex-determining gene arose, and which then deemed the nascent Y, which was X at the time. And what happened is that the genes moved off of the Y chromosome, such that we get what we call heteromorphic sex chromosomes. And you're right, the Y chromosome has, and I don't know what the number is, maybe there's probably somebody in here who knows better than me, but it's 4 or 5 genes. The main one is this thing called SRY. But it sits upstream and then dictates that individual, if it positively has the Y chromosome, that all the cells are going to be male in that organism if it works right. And the absence of which is XX, right? So sperm either has an X or a Y. Will it continue to degrade? I think it will degrade to the point where it potentially could have just the SRY gene on it. I think that's possible. Yeah, I don't know what. I mean, there's going to be. We're sort of stuck in this sort of two-sex system in our species and in other species, and so the selection against that breaking down will probably be strong. So that will be probably. I mean, I guess that would be maintained, but it would get down to the lowest possible situation, which is the lowest number of genes it could carry, and still determine that zygote to be an XY zygote, and thus a male. Yeah.
As I mentioned my little mission list last night, this morning, those of us on this campus consider a little building across the street one of the cradles of discovery of sex and bacteria. How do people in your field feel about using the phrase sex and bacteria compared to the meiosis field?
Good question. My friend Nicole's here, so some people really don't like it. So some people only want you, when you're talking about sex, to be talking about meiosis. I'm a little more flexible. I've actually worked on prokaryotes and eukaryotes throughout my career, so I'm a little more flexible on the issue. The logic behind the people that really don't want you to use sex when you're talking about bacterial exchange is that mechanistically and phenomenologically, it has both different ways of doing it and different outcomes. And whereas meiosis really is this all-in phenomenon. Every gene is shuffled, right? And whereas bacterial sex is one or a few genes at a time. I liken bacterial sex to kind of like Pokemon card trading. That might not work at this audience either. But baseball card trading, how's that? Is that you can trade one or a few at a time to increase the value of your genome. And that's what bacteria do, and it's a really great way to do it because you got many, many individuals, you got many, many combinations. Bacteria also don't tend to allow duplications of genes in their genomes. There are lots of things about prokaryotic genome evolution that really only allows sort of a few things at a time. So the origin of eukaryotes had something to do with this difference in the way that genetic variation can be parsed between individuals. And did it require the whole deck at a time? Maybe. It certainly the few genes at a time works very well for bacteria. They swap genes all the time. Do I want to call that sex? I'm okay calling it sex. But I think the distinction should be made about all in or a few at a time. That's sort of the way I think about it.
I thought the other thing was interesting, and Millard Sussman made this comment Monday when he was talking about Letterberg's. He gave a talk here.
Yep, which I wasn't able to go to, unfortunately.
It's online if you want to.
Oh, good.
I think Letterberg, correct me if I'm wrong, but Letterberg was arguing at one time that the two bacteria were actually fusing, kind of like gametes. And another guy was saying, no, it's just being transferred. And if it had been two bacterial cells fusing, it'd be a lot more like meiosis, but then you have this.
Yeah, people have looked for that, like in archaebacteria, as a kind of presaging for eukaryotes. I don't know about that. Nicole, do you have think? Was that an artifact, or was? I mean,
it was a hypothesis.
Oh, so nobody ever saw it. Just if that was the way it worked.
Letterberg was arguing for it as a hypothesis of how you would go about testing it.
Yeah, I mean, bacterial cells don't tend to do that, right? I mean, archaebacterial cells have blebs, and they sometimes touch each other. It has to do with their outer membranes and such. I don't know that much about that biology. I mean, even if they did fuse, the way I imagine it is that one genome or the other one would win. It wouldn't be that they would come together and be happy, probably. Would you agree with that?
Yeah, but I don't think we've got to see that.
Yeah, because they tend to not tolerate duplication. They prefer to have a single genome because they're, and that's why sort of one or a few genes at a time works pretty well, because you can swap them in and out as cassettes. That's a cool line of thought. I should go back and look at that paper. Yep.
Oh, okay. So you talked about sex being able to bring together useful mutations, but isn't it just as likely to be able to split them apart?
Yep, absolutely.
Cool. So.
Yep, absolutely. That's also in the bad. It's actually not one of the things I mentioned, but it actually breaking apart adaptive combinations of genes is another good thing, right? So the analogy to me was, maybe I've got two genes next to each other on a chromosome that are really good. Well, if I had a recombination, then none of my kids are going to get them both at the same time. Yeah, excellent question. Yeah.
First, a clarification question. The work that was done was there was a bunch of organisms that underwent meiosis, and a bunch of genes were looked at, and then we wanted to see which one of these genes were in each one of these organisms, right?
Correct.
When you did that, was there a pattern that appeared for the condition of meiosis? Because clearly it's not the number of genes that are present, because you showed us one with just 4.
Yeah. There's nothing to, so one of the neat things about meiosis, if you look at it in lots of different organisms, on the surface, it looks pretty much the same. I mean, the effects, I mean, there's always chromosomes that are pairing by this thing called the synaptimal complex. It might vary in, you know, in its length or something like that, but if you can get it, if you can visualize what's going on, it looks like it's carrying out the same process. So there's nothing discernible to my eye, although I can tell you a few cases where I know something more. But in general, just because it's missing this or that gene, I don't have a clear idea of what that means in general across, you know, things that are missing those genes in common. Let's say that's what you're kind of interested in. Is there some shared feature of meiosis being weird by virtue of missing those two genes?
Or just if there was like a condition that was, like if I saw just certain genes that appeared, can I say for certain or not for certain that this organism might undergo meiosis?
Yeah, no. I'm making a general form of an argument. I'm saying if they've got, and so I sometimes say it as if they've got the goods, they're doing the deed, but what do I define as the goods? Some of the genes. Okay. And what's the deed? Meiosis of some flavor that I can't really, that might be slightly different.
And has the work been looked at the other way around, where we looked at organisms that do not undergo meiosis and we look at the gene distribution?
Yeah, so that's like what we tried to do in the Dalhousie rotifers, and I had another example that I could have shown today, but so we're finding some cases where things that we think are asexual, and we're finding some degradation of the meiotic genes. So yeah, I didn't get to talk about that tonight, but we're finding some evidence of that as well. But it's tricky because you got to catch them right in the act of losing them, because once the meiosis genes are degrading, maybe that means that the organism themselves are going to go extinct because of this idea that asexuals don't live very long. So so yeah, that's a tricky part, and part of what we've been exploring. Yeah. Those are great questions. One more.
Yeah. In talking about the meiosis species and the asexual species, you're saying, I think you said that the asexuals are failures.
Yeah.
But how can that be? Because they're reproducing, you know, fast.
Yeah. So let me define. Yeah. Let me define failure. The failure means that once they arise as an asexual lineage, they tend to not persist for a really long time, millions of years. So you don't find lineages that you can demonstrate are asexual. That's why Dalhousie rotifers were so interesting, because they had persisted as a group for 100 million years, and yet ostensibly they were asexual. So if true, they become the poster child for how to make it work in the absence of sex. And they still may end up being the poster child, and maybe they have figured out some mechanism. But I'm just a little bit skeptical of being able to say right now, given the data. And they were kind of the most extreme example of a lineage that was well documented by lots of people, and they never found any sexuals among those Dalhousie rotifers. Still could turn out that that's the poster child, and really, well, and that's why they're really important. They could be the exception to help understand the rule.
All right, thank you very much.
Thank you.